Welcome to Mind Over Matter, where we feature young Jamaicans who are shooting for the stars. I'm your host, Margaret Boyne. Lung cancer remains a devastating disease. Today, my guest is a young researcher whose work on small cell lung cancer has the potential to revolutionize treatment. He's a 24-year-old student at Stanford University pursuing a Bachelor of Science in Biology and a Master of Science in Epidemiology and Clinical Research. He's an undergraduate researcher at Stanford University School of Medicine. A Gates Scholar, he's a recipient of the Stanford Cancer Institute Undergraduate Fellowship Award, the 2023 Prime Minister's National Youth Award for Excellence, and a 2023 Rhodes Scholarship finalist. And these are just a few of his awards over the years. He shares his academic journey from Ardenne High School to Stanford University and his groundbreaking research on small cell lung cancer. My guest is Kyle Lambert. Welcome to Mind Over Matter, Kyle.
Thank you. I'm very, very excited to be here.
And it's a pleasure. I'm looking forward to talking to you, especially about the work that you've been doing at Stanford. You have been doing some remarkable research at Stanford University at only 22 years old. So let's talk about your work on small cell lung cancer. What specifically are you investigating? And you can break that down for us.
Yes, certainly. So I'll start with an overview of what small cell lung cancer is. So this is, as you can tell from the name, it's a cancer that it's site specific to the lung, right? That's where you have the primary, what we call primary tumor. On the other hand, small cell lung cancer is a type of cancer that metastasizes a lot. So what this means is that it spreads to different sites. So while the primary site, the place it initiates, the place it starts is in the lung, most of the patients, by the time that they actually present in the clinic, they have tumors in the liver, they have tumors in the brain, tumors in the kidney, and that's just the type of aggression that you see with small cell lung cancer. So the survival rate is very low, right? For a 5-year survival rate, which just means in 5 years, how many patients actually survive? 7%. So it's one of the most deadly cancers. It's typically associated with smoking. So most of the patients, when they're presenting in clinic, they have lots of lots of years in which they have been smoking cigarettes, right? And what I have been looking at is the why and the how behind these cancers. So a lot of times when we think about these big picture diseases, we oftentimes, it can be a bit scary because there's a lot of things to uncover behind how these things are actually happening. What causes one person to have cancer in this area and another person to not have it, right? So on the most basic level, we're looking at cells, right? There are trillions of cells in our body. Actually, it ranges from 18 trillion to, I believe, about 28 trillion in men, the amount of just cells in your body. And what's actually happening is that you have these changes, even these minute changes, these small changes in one cell that can cause it to start proliferating faster. It starts growing faster. And that's what actually characterizes cancer. My work focused on understanding what is actually happening inside the cell that allows it to then grow faster, to proliferate so much, and to the point where it's actually leaving the lung and entering distant sites, or it's entering the liver, it's entering the brain. And that has just been so exciting. I've looked at it in a genetic level, which I'm happy to talk about a little bit more, how we look on the DNA, how we look on RNA, proteins, and kind of walk through that. But what I really looked into was the mechanisms of these metastases, the mechanisms of how it's actually moving from one place to another and uncovering specifically how the genes are doing that. And outside of that, I've looked into another set of diseases, paraneoplastic neurologic syndromes. Big words, a lot of jargon in that. Trust me, it took me some time to even be able to kind of say it, right? So these diseases are related to small cell lung cancer, actually, because what's happening is the cancer starts what we call expressing. So it starts to have proteins that it doesn't typically have. Actually, it starts to express proteins that are only found in the brain. So these tumors in the lung are actually showing proteins that you would only find in your brain and in the nervous system. So the immune system gets confused here. The immune system is coming to attack the cancer, right? And the way it attacks the cancer is actually looks for the proteins and say, okay, I got you now. I find this protein. I know exactly what to look for to attack you. However, what else expresses that protein? The brain.
Brain,
So what the
yes.
immune system is doing is not just attacking the tumor, but it starts attacking the brain as well because it thinks the brain is foreign because it has these proteins. So these patients come in with a lot of neurologic symptoms and these subsequent diseases that, only takes their cancer even further because now you're dealing with the cancer and you're dealing with these neurologic symptoms. So it really, really becomes tricky, and it's been exciting to kind of understand how these things are happening, because by understanding how they're happening, that's how we can develop treatment approaches, right? You can't treat something you don't understand. And so my work has been a lot of that figuring out how it is actually happening to allow us to start thinking about treatment strategies.
So why is it so hard to treat though, or to find a cure for lung cancer?
Right. So that's a great question. The big issue is that cancer is something that's always changing. So remember that those genes that I was mentioning and what we like to call like cancer dynamics. When you figure out what gene is actually allowing these cancers to metastasize in one case, when you look at another patient, you're going to find something different. It is
so totally
vastly,
different.
exactly. You have these vast differences that you find between patients that it becomes tricky to say, okay, let's come up with a cure. And that's why sometimes when a lot of people say, when we're going to find a cure for cancer, when we're going to find a cure for cancer, it really is a lot more difficult than that because, to scientists, to doctors, when we think about cancer, it isn't just one thing. It isn't just one thing that I have to treat. What you have to treat is this specific kind of cancer. This patient expresses this one protein that we can kind of use to attack, but this other patient has another one. So, to come up with these treatment strategies, you have to account for the big differences that all these patients are happening. So that's why it's important to have the science behind it, right? You need scientists who are saying, actually, when we look into these patients, we're seeing a high amount of patients expressing this kind of protein. So maybe what we can look into is treating, let's say, 10% of the patients who have this specific protein, and that's a treatment strategy that we have for them. So it becomes an issue of, not how we can treat cancer as a whole, but how can we treat patients with small cell lung cancer who has this protein expression? How can we treat other patients who have this specific protein expression? So that you have what we call personalized treatments. We have, the treatments that are specific to people. And that's the future of cancer. It's the current and it's the future of cancer research, where we're looking at how to tailor a treatment approach to the patient. And that's how we get higher success rate.
So what you're saying then, it's not a one size fit all cure.
Correct.
It's going to
Right.
have to be a personalized one.
Exactly. We have to have these answers that are looking at the patient-specific tumor. And it becomes a lot easier because that's daunting, of course. You're looking at several patients, if you have 10 patients in a room and all of them have different gene expressions, and it's like, okay, how are you treating all of these? It's one, expensive, right? It's expensive to come up with these drug approaches. It's expensive to have the resources to even develop them. But what you realize is through science, if I am if I have these patients' tumors that I collected and I am now doing my research on it and I'm seeing, okay, here's a way how I can treat this. Another year down the line, another patient will come with that similar type of expression. And then another patient will come with that similar type of expression again, right? So what happens is that the more we build this toolkit of treatment approaches, the easier it gets for future patients and the less likely that a patient will come and say and show us a tumor that has a different expression that we haven't seen before, right? So as we build this toolkit and we're learning from different tumors and learning what's actually being expressed, it becomes easier for patients down the line.
But I've read, Kyla, lung cancer is the leading cause of death. but yet
Right.
still I hear more about breast cancer. What are those
Right.
other cancers? So, I mean, why am I not hearing more about lung cancer cures then or
Right. Yeah.
treatments?
So that's the thing. So it's what you'll hear more about. What you'll hear more about are the cancers that you have more, I want to say, promising treatment approaches. When it go into
Okay.
clinical trial, you're seeing a
Mm.
lot more success rate. In breast cancer, there are a lot of researchers working on that. Meanwhile, small cell lung cancer, it's extremely daunting just because of
Mhm.
how deadly this cancer is, and it's so intricate in the sense that understanding the molecular side of it becomes extremely challenging. And those challenges do persist in breast cancer and colon cancer, pancreatic cancers, the cancers you might hear about more often. But what happens in science is that it is a bit more difficult to deal with a cancer where the patient presents in the clinic, and unfortunately, because of the aggression of the cancer, they're only living for 7 months, 8 months after the time of diagnosis, right? As opposed to other cancers where you're able to follow the patients for even a year, or 2 years, 3 years. And what you get from that is you get better data. You get to understand the tumors better. You get to understand the effect of the tumors of the person better. With small cell lung cancer, we don't have that on a human level. So what we
Mm.
actually rely on is
Oh.
creating and engineering mouse models, right?
Mm.
So that's something that I have worked on a lot, and learning how to, have a mouse and actually allow it to have the kind of cancer that we see in humans, to replicate the kind of cancer so that we can slow down the process and understand at each time point what is actually happening, what genes are being expressed, what changes in the different proteins are happening. And that level of work becomes a lot slower. So you see a lot slower time points of getting to the treatment approaches, but it does allow for a more robust understanding. We have a really rich understanding of the molecular intricacies of small cell lung cancer that we're working on. And I'm very, very hopeful for the treatment approaches as we're building on that. And the science is getting just so much more interesting and so much larger right now, so that we can see that same level of success that we might be seeing in other cancers.
Are there any significant findings you've been having so far that you can mention?
Yes, certainly. So a recent paper that I published in collaboration with Dr. Julie Cole, she's a cancer biologist who trained at Stanford that is now working at UCLA. We worked on this transcription factor, and I'll talk about what a transcription factor is, NF1B. So in cancer and in all your cells, we have three big words that it's a simple crash course in how these things happen. So our genes, right? Everybody knows that, in our body, in our cells, we have DNA. That's what we call our gene. Those are the things that it has all the code, all the things that we use to actually define the color on our skin, the shape that our eyes are going to get, the the okay, congratulations. The decisions on, when we're tired, all these things are happening from our gene. So, but how? There's this beautiful machinery that we have in our bodies where the DNA is interpreted, it's read and translated, or transcribed, I should say, sorry, into what we call RNA, right? So you have these different things in the body in the cells that read the DNA and say, okay, what do we need right now? We're tired. It's time to go to bed. Let's take out what we need from the DNA and make some RNA so that the person can be sleepy. Same thing for the skin color. Same thing for the cancer. When the cancer decides it's hungry and need certain things, it's transcribing DNA. Okay, so now we have this RNA process that actually has the code for what the cell needs in the moment. So what it does is it sends to these really cool machinery in our cells that we call ribosomes, right? And these ribosomes change the RNA into proteins. So now we went from DNA to RNA to proteins. And these proteins are the actual functional things that we have in the cells. So the melanin that we're seeing, the things that are causing your eyes to blink when you blink, those are all proteins. Those are the actual functional side of things, right? So what I looked into in my work was how the transcription factor NF1B, which means this is a protein that binds to DNA, right? It binds to DNA and it says, I want this part of the DNA to increase in expression, right? Which is, for majority of what our cell needs, it works, right? That's why most people don't have cancer. It works.
Right.
It's a beautiful machinery that really does, show how complex the body is. However, what NF1B does in cancer is it actually opens up the DNA a bit more, because as you can imagine, our cells are tiny, tiny, tiny structures. Yet each of our cells have so much DNA, so many things that they have to actually, decide, okay, at this point in time of the day, this is how we're going to work. It's a lot of information in our cells. So usually how we have it is it's very, very, very compact. It's a very compact string of just these molecules that we kind of have tight inside of our cells. NF1B opens up that DNA so that it has access to kind of transcribe and become RNA, right? So what we realize is that NF1B, by opening up the DNA in that way, what it's actually doing is increasing expression of genes that are what we call oncogenic, that are actually boosting proliferation and increasing the chances for the cancer to move from the lung to other parts of the body, right? Our big finding there was that there's what we call NF1B switch. So when NF1B is working and in the cells, it's the main driver of metastasis. It's what's actually driving the progression from moving from the lung to another site in the body. What we also found that scientists have never shown before was what actually happens when NF1B is low in the cell, though, because, as I mentioned earlier, patients present with all sorts of expression. Some people have high NF1B, some people have low NF1B. So how comes people with low NF1B still have metastasis, right?
Right.
If NF1B is the big driver, then how are they still having metastasis? What we found is that, just as I was saying earlier, cancer is dynamic. It's going to find another way, right? So it uses another gene, another transcription factor called FOXA1, where it uses that now when NF1B is off to actually drive metastasis. But while this is daunting and, we can look at it and say, boy, this cancer, it always going to find a way. We're learning from it now and saying, okay, so in patients that have low NF1B, can we have a therapeutic strategy that targets FOXA1, right? So that's how we learn from these things. And, even though it becomes a huge web of all these transcription factors and proteins that we now have to consider, from this paper that we published, we saw that there's another avenue. There's something else that we can look into for therapeutic strategy. And that's always exciting, right? So that's one finding that I've recently looked at. I also just put out my work under the Stanford Digital Repository, actually, that looked at the paraneoplastic neurologic syndrome that I was talking about earlier. What I did there was actually figure out the mechanism by which these happen in small cell lung cancer. So another set of genes called the HU protein, also called ELAV-L proteins, they work in the intricate network to kind of allow for that neuronal expression on the tumor that you wouldn't find anywhere else in the body but the brain. And what I kind of did there was to look at the actual mechanism by which that happens, which hasn't been looked at before, and provided some novel insights, some new insights on how we can actually treat these patients. I found that the gene ELAV-L2 is actually a key player in allowing for the progression of paraneoplastic neurologic syndromes, which was never proven before.
Very interesting, Kyle. We hear a lot about AI and how it's gonna transform the medical care and healthcare. So are there any areas where, AI could complement your research?
Certainly. I have been a big fan of using AI, especially in the context of we have a lot of data. When we're looking at the human genome, when we're looking at all this RNA stuff that I was telling you about, it is a lot to parse through. It's a lot of information for one person to sit down and go, okay, let me try find this gene. How much is this gene being expressed? Because in the, back in the days, what they would have to do is they sit on a computer, you sit there, you look through all the genes, look through all the DNA, look through all the RNA. I'm not doing that anymore, right? That's not something that I have to spend my time doing. I can spend my time now thinking about the thorough experimental question, because through AI, what I've been able to say, okay, come up with some code that I can use to parse through this data and figure out which genes are being highly expressed. So now instead of spending 2 hours trying to look through all this, sifting through this data, I have artificial intelligence that I can show this data, and it can say, okay, here is the 3 big genes that I'm seeing being expressed. So you turn 2 hours into 10 minutes. What that allows scientists and doctors to do is to spend more time on the things that are, a bit more difficult, things that AI can't necessarily do, right? Because you can present AI with these complex things, and it can sift through it and maybe has a better eye than us in finding something. But it can't think. It can't think with several pieces of information and synthesize it the way a scientist and a doctor could do, right? So that's the way I see it helping us right now and in the future. I believe that especially as we think about cancer, it is an amazing tool. And what I would, I definitely want for people to understand that a bit more, how, though it's something that we definitely should have, laws and regulations around so that it's being used appropriately, it is an important tool that, I want people to understand how scientists, how doctors are using it in a way to come up with those treatments faster, right?
Better outcomes.
Much better outcomes, and actually having space to focus on the things that are a bit more difficult that AI can't necessarily look into.
You received funding from Stanford School of Medicine,
right?
For your research, and I read that this is very fascinating because funding is usually reserved for persons who, have graduate degrees. Tell us
Right.
about that.
Certainly, yes. So I have been so blessed to be among mentors who have guided me in this path and allowed me to have, a level of science that I am proud of, and a level of science that, has transformed a lot of the ways I think of biology and how I can practically use biology. When I was in my second year of college here at Stanford, I talked to my boss, my principal investigator, and said, I have this idea. And I was reading this book, and I learned about these diseases called paraneoplastic neurologic syndromes. And it's so interesting how a cancer can be in the lung, yet the immune system is having these aberrant responses that cause damage to the brain. And what he said was, take the initiative, write about it, see how you can see what kind of funding you can acquire from that. So I took the big step of applying to this major grant here that we have called the Translational Research and Applied Medicine Grant. And as you stated, it is primarily for experts in the field who already have their medical degrees, already have their doctorates, and have extensive work in cancer biology to know, come up with what we call a pilot study to start off something that has never been looked at before. So when I applied for that, while nervous, I was excited because, I knew I had the foundation. I knew I was building a lot of the cancer biology techniques that I needed from the same NF1B project in small cell lung cancer that I was telling you about. And it worked, right? So I was successfully awarded funding for that and received just amazing mentorship under Dr. Dean Felshaw, who is the director of that program, and as well as the chief of the MD, PhD training program we have here at Stanford. And it was exciting kind of just being around those experts as someone who is 22 years old, being around these people with decades of experience in this field. It was good because you kind of bring this novelty to the field where, I am way more fascinated with these new age techniques. I am more fascinated with the more new things that I can bring to the table, while also learning from these people who have these vast experiences that can fill my cup that has been built here at Stanford so much. And so that's been an amazing experience, and also just being recognized on that level has been great. And what it has allowed me to do on the research side of things, and building from there, it has just been truly amazing. I've been so excited to propel that work and utilize what they have given me to further the work that we do in cancer biology.
Stanford is one of the most prestigious research universities, but you were accepted at six. You're accepted at 6 universities, including Yale. Was it a difficult decision to choose Stanford?
Oh my goodness. It was a very difficult decision because,
And let me say 6 Ivy League.
right. Right. It was a difficult decision, I miss when I had moved to the US for that last bit of high school, because I was here for 2 years. And I was like, okay, it's time to kind of decide what kind of schools I want to go to. And, having, of course, the education that we get in high school in Jamaica, the preparation is amazing. And I tell people that all the time.